Fabiola for her hospitality. My name is Kimberly Norman, and I'll be speaking on integrated testing strategies for skin sensitization prediction. I work at the Institute for In Vitro Sciences, which is just in the Washington, D.C. area. And in our lab, we specialize in non-animal methods for toxicology testing. Skin sensitization, or allergic contact dermatitis, is the most common occupational health-related adverse response, with a cost of greater than $1 million per year. Therefore, it is a critical endpoint in the evaluation of new chemicals, as hundreds or thousands of chemicals are implicated as skin sensitizers. Skin sensitization must be assessed for all substances registered under REACH and the FDA. And per the 7th Amendment to the Cosmetics Directive in Europe, there is a ban on animal testing for all cosmetics products and ingredients. And that also applies to any company selling cosmetics or ingredients in Europe. Here's a brief cartoon on the mechanism of skin sensitization. Beginning here in the top corner, with a chemical or hapten, if it's able to penetrate the skin, it can interact with the cells in the epidermis. When it's recognized by these immune cells of the skin, which are the Langerhans cells, it can migrate from the skin into the draining lymph node. At the lymph node, these mature Langerhans cells present the chemical to the T cells. This causes the proliferation of specific T cells, which are then released back into the systemic circulation. And upon subsequent skin contact with the chemical, it's going to activate those T cells and lead to the clinical manifestation of allergic contact dermatitis. And here are some of the clinical pictures of allergic contact dermatitis. It can be caused by many things. One picture here is for, there may be contaminants and dyes like henna. Henna itself is not a sensitizer, but there may be contaminants. Things in makeup or cosmetics may cause sensitization. Here on the neck would likely be perfume. And along the ear, there are certain metals that cause sensitization, such as nickel, which is commonly found in jewelry. So just a little background on the evolution of this endpoint and how it's been evaluated over the years. In the 1970s, the guinea pig test methods were developed. And then moving into the 1990s, the assay was refined such that the number of animals required to evaluate the endpoint was reduced, and the animals would experience less pain if there were a positive response. That's called the local lymph node assay. Both the guinea pig assay and the local lymph node assay are still used. But now, as we're moving into 2010 and beyond, there is significant focus on in silico, in chemico, and in vitro methods to address this endpoint so that we can reduce the amount of animals that are used for this testing. There's also testing on human volunteers called human patch testing. In my talk, I'll be focusing on the in chemico in vitro methods. So Europe has been leading the validation for these non-animal methods. There are 3 methods that are currently at an advanced stage of revalidation, and they are the human cell line activation test, or HCLAT. This was developed by Kao Corporation in Japan. The direct peptide reactivity assay, or DPRA, which was developed by Frank Gerber and colleagues at Procter & Gamble in Cincinnati, and the keratinescence assay, which was developed by Andreas Notch and colleagues at Givaudan, which is based in Switzerland. During the evaluation of these assays by ECVAM, they have concluded that each assay is considered adequately reproducible to be considered for an inclusion in an integrated testing strategy for hazard identification. Because the animal test is very comprehensive, it's going to be challenging for any individual assay to replace completely the animal test. So the short-term goals now for ECVAM have been to develop an adverse outcome pathway-based integrated testing strategy for hazard identification, and to work on the development of OECD test guidelines on these 3 methods so that they may be used in a regulatory context. So here's just a little information on the adverse outcome pathway for skin sensitization. And the adverse outcome pathway is just a theoretical framework for how you could address the critical points of this pathway towards this physical manifestation. Beginning with looking at the molecular properties, what is the molecular initiating event, what is the cellular response, the organ response, and finally the organism response. For sensitization, we know that you need to penetrate, the chemical needs to penetrate into the viable epidermis. And then one critical marker for sensitizers is their electrophilicity, or their potential to be transformed into electrophilic chemicals. So with that electrophilicity, you're going to have this covalent interaction with proteins. This in turn is going to cause the expression of certain cell surface markers and cytokines, thereby leading to this proliferation of T cells in the lymph nodes. And then lastly, you're going to have this organism response, which we can see here, of the redness and swelling. So I'm just going to give a little information on each of these assays. The human cell line activation test, or HCLAT. As I mentioned, these Langerhans cells play a critical role in skin sensitization. Upon antigen capture, the Langerhans cells undergo maturation and migrate to the draining lymph node. This Langerhans cell maturation is characterized by the upregulation of CD86 and CD54. So in this assay, THP1 cells are cultured with a range of doses of chemical, and it's allowed to incubate for approximately 24 hours. And then the cells are stained with these cell surface markers, and cell viability is measured by flow cytometry. Therefore, those chemicals which cause the increased expression of CD86 and CD54 could be identified as sensitizers. Next, the direct peptide reactivity assay. This is considered an in chemical assay because no cells are actually used. You're just simply looking at the reactivity of two chemicals. So the correlation of skin protein reactivity and skin sensitization is well established. So in this assay, you just take your chemical allergen and then you would incubate it with these short model peptides, which would contain either a cysteine or lysine, and you would allow them to interact for 24 hours and then just monitor depletion of the peptide by HPLC. And the more depletion you observe of the peptide, that would indicate a higher reactivity and a sensitization potential. Lastly, the keratinocytes assay. Once again, a feature that sensitizers have in common is their intrinsic electrophilicity. And this pathway, the NRF2 electrophile sensing pathway, is able to detect this electrophilicity and thereby skin sensitizers. So it is composed of this key repressor protein, and when an electrophilic skin sensitizer interacts with the cysteines on key, this causes a covalent modification of key, thereby allowing the dissociation of the transcription factor NRF2 from key. NRF2 is then able to accumulate in the nucleus where it causes the induction of genes with an ARE in the promoter sequence, such as the NRF2 target genes. So in order to use that pathway and create a simple laboratory-based assay, Givaudan began with a packet cell line, which is an immortalized keratinocyte cell line. Into that, they engineered a reporter construct with a copy of the ARE element of the human aldoketo reductase gene upstream of a luciferase gene. So in this assay, it's a simple endpoint where, once again, you would have your chemical, if it interacts with those cysteines on key, that would allow dissociation of NRF2, and you would cause induction of your luciferase gene, which can be easily monitored in the lab. So if we think about these various assays in terms of this adverse outcome pathway and how we can model the key events of sensitization, we could consider that the direct peptide reactivity assay is monitoring protein reactivity. The keratinocyte assay, or keratinocytes assay, is monitoring keratinocyte activation. And the HCLAT assay is evaluating this Langerhans cell activation. Now I'm just going to go into a few specifics of the DPRA assay. So in terms of the laboratory steps, you would simply prepare your HPLC machine and your reagents, test articles, and control solutions. As I mentioned, the test articles are evaluated by HPLC following the 24-hour reaction period, and the samples are run in triplicate, and timing, of course, is important. And then the chromatograms and peak area data for each test article and reference control is generated. For the assay controls, each assay is run with both peptides, short model peptide containing lysine and short model peptide containing cysteine to assess HPLC system suitability. And the positive control is dynamic aldehyde, which is a non-sensitizer. One of the limitations of the assay is that the chemical must be soluble. So acetonitrile and water are the preferred solvents, but you may use many other solvents and combinations of solvents to achieve solubility. And of course, you want to avoid precipitate if possible, because this could damage your HPLC machine and certainly be challenging to interpret those data. Another concern with the assay are those test chemicals which would have the same retention rate as your model peptides. In that case, the percent depletion cannot be measured, and so you would need to ideally use a different assay for those chemicals. And for the data analysis, here's just a simple image of your unreacted peptide, your test chemical, and then your reaction mixture. And what we're looking for is the peak area of your unreacted peptide as compared to your peak area of your reactive peptide to indicate the reactivity and, in part, sensitization potential. For the prediction model for DPRA, this text is a bit small, but basically you look for your percentage of depletion for both cysteine and lysine, and then you can rank it from minimal reactivity up to high reactivity. And below you can see in yellow or orange, you can then classify those reactivities into non-sensitizers or potential sensitizers. As just a summary of this method, the advantage is that the data can be accumulated very rapidly in a couple of days. It's a very cost-efficient assay, and as compared to the existing animal data for the chemicals that have been evaluated, it's about 80 to 85% accurate. The limitations are that the chemicals must be soluble. This is certainly a challenge with things like the botanical extracts. Coelution is possible, and it may not detect pre- and prohaptins, or those chemicals which require metabolic activation or oxidation to exert their sensitization effects. Next, I'll move on to the keratinosense assay. This assay, as I demonstrated earlier, examines a cellular toxicity pathway that is induced by skin sensitizers. It's a novel keratinocyte-based reporter cell line, which was developed by Gemini. It's a 96-well-based assay with a technically simple format, and an interlab study has been completed, and the results have been published. These results indicate a good predictive value, and so the assay has continued to be successful through the years. Just some information on how you actually conduct the keratinosense assay. These cells are seeded into 96-well plates. They're allowed to attach overnight, and then the following day, your test chemicals are dissolved in either DMSO or water. And then this is serially diluted, your test chemical, in a range from approximately 1 micromolar to 2,000 micromolar across your plate. And here's just an example of a plate where you can run up to 7 chemicals on one plate. Each plate will also contain your solvent controls and your positive control, which is also dynamic aldehyde, and blank well. And for each assay, you're going to run each chemical in 3 independent trials, and each independent trial is performed in triplicate. So you're generating a substantial amount of data on each chemical, so you would have confidence in your ultimate answer. So once the test chemical is placed on the cells, it's incubated for approximately 48 hours, after which the cells are lysed and luciferase substrate added. In separate plates, we also evaluate cell viability using the common markers MTT and MRU. So then moving on to the data analysis. We are comparing the gene induction of that luciferase gene to the DMSO control well luciferase gene induction. We found that the critical threshold is a 50% enhanced gene activity. This has been coined the EC 1.5, or the concentration in micromolar for induction above this 1.5 threshold. We also evaluate the Imax, which is the maximum fold induction, and a cell viability endpoint. Here in the graphs below, these are just representative graphs of a non-sensitizer and a sensitizer. Here you can see I've highlighted that 1.5 threshold, and in the dark blue, you'll see that's the induction of the luciferase gene, and for the non-sensitizer, it never crosses that red line, so that would indicate that it's not inducing the luciferase gene. And in pink above, it also has no effect on cell viability. Whereas for the sensitizer, you can see that there is a sharp induction crossing the red line, our EC 1.5 value. Also, in this case, the Imax is about 5. And it also, using the pink line as a guide, you can see that it also has a concomitant effect on cell viability. And so just a little more information on how we use this data to identify a sensitizer. For a material to be identified as a sensitizer, the EC 1.5 value must be below 1,000 micromolar in at least 2 of the 3 independent trials. At the lowest concentration with a gene induction above 1.5, the cellular viability should be above 70%, and there should be an apparent overall dose response, which is similar between repetitions, which we assess by statistics. So our lab participated in an interlaboratory study of the keratinosense assay. The European Center for Validation of Alternative Methods, or other validation or national authorities, will often fund these projects to evaluate the methods. In this case, we were evaluating transferability, the intra- and intra-laboratory reproducibility, and predictivity of the assay. We were comparing using the existing animal data on this set of chemicals to evaluate predictivity. In this interlab study, Givaudan from Switzerland participated, which was the lead lab, BAS and Bayersdorf from Germany, and Procter & Gamble and IIBS from the United States. And in this study, we evaluated 28 chemicals in 2 phases. So here is just an example of some of the published data from that study. Here we see all of the lab's data for one of the sensitizers. This is a moderate sensitizer, Citral, and you can see that for each of the labs, you see a very similar dose response and a very sharp peak in the luciferase gene induction in dark blue, and you also see cell death demonstrated in the peak. And here's an example of data from a non-sensitizer, sulfanilamide. And the EC 1.5 is just highlighted with that black line in the middle. And you can see that for each of the labs, the luciferase gene induction never crossed that line, nor was there any effect on cell viability. So between the labs, we had very reproducible results. This is a compilation of all of the data that we generated as part of the ring trial. I'll try to go through it briefly. So in the top portion, you'll see that these are the sensitizers, and in the bottom portion is the non-sensitizers. You can use the colors to see how well the assay performed. So in the top portion, in red, 3 of 3 would mean that in all 3 independent trials, the assay was identified correctly as a sensitizer. And as we see for most of the sensitizers, this was the case. However, for certain sensitizers, I'll give the example of eugenol, it was not predicted correctly in each independent trial or in each lab. So there were some discrepancies. And then for the non-sensitizers, generally these were predicted correctly in each lab. So as a wrap-up of this interlaboratory study, the assay was found to be transferable, reproducible, and predictive across laboratories with a predictivity of about 86 to 96% as compared to the existing animal data. The EC 1.5 value, the variability, was low both between lab and within lab, and the IC50, or the cell viability measurement, was similar. We found the assay to be cost-effective and time-efficient, and the data were published in 2011. Because of the success of the ring trial at IIBS, we began offering the service commercially in 2011. And since the assay was developed by Givaudan, which is a large fragrance house, a large portion of the materials that have been evaluated are fragrances. And the assay really needs to be further tested to understand the applicability domain. So we can perform the research services, and we encourage sharing of non-confidential data so that we can share it in forums like this, so we can share with others how the assay is performing for those types of materials. We've been evaluating both ingredients and mixtures, including preservatives, surfactants, solvents, botanical extracts, essential oils, and even medical device extracts. So now that I've discussed a couple of the assays, I'm going to go back into how do we put this information together in order to replace the animal-based studies. So I'll just remind you that through ECMAM, they have concluded that each assay is adequate to be considered for inclusion in an integrated testing strategy. They have not said which integrated testing strategy should be used, but they say that you should use one. And right now they're working on determining how you should use these assays in combination or in a tiered approach. So, also, once again, there's many ways of looking at how you should put together these pieces of information. If you take an AOP-based approach, you would think, okay, I need to have an assay which monitors each critical step of sensitization. However, if you just take a data-based approach and see what your predictivity is, you may come to a different conclusion. So I'll just give a couple quick examples here of groups that have been determining which integrated testing strategies will be most accurate. This is a colleague's data from BASF, and in the top portion, in orange, these are all sensitizers, and these were evaluated in about 5 different in vitro assays. And also the human data and the LLNA data is listed in this table. Those areas where you have an inaccurate prediction is circled in red. And the bottom portion here are the non-sensitizers, and we have some incorrect predictions for those also. Okay, so then if you take this information and you try to determine what's the predictivity of each assay alone and what's the predictivity of the assays in combination. So here are the assays, the individual assays, DPRA, the keratinogens, and the U937, which is similar to the H-class. And if you compare the data to the existing human data, they're all quite accurate, but they're all very similar in their accuracy. If you compare it to the animal data, the accuracy is a bit lower. And in the second column here, I have the accuracy of 54 chemicals compared to LLNA. And in the second one, or the last column, it's 145 chemicals, which were evaluated in a larger study. But the numbers are essentially the same. So if you look at the bottom row, if you take an approach, a weight of evidence approach, and say if 2 of 3 of the assays are either positive or negative, that will be my classification of a sensitizer or non-sensitizer. When that was done, as compared to the human data, it was a 94% accuracy. So that's very powerful evidence that this approach may be used to accurately predict skin sensitization and be a full replacement for the animal test. Lastly, there are always considerations when replacing the animal test. For instance, these assays do not address skin penetration and skin metabolism. Therefore, you could have some over-predictions or false positives. Solubility is always a concern. Certainly, we know in the real world every chemical is not soluble, So perhaps moving into a three-dimensional cell-based model could help address those types of materials. These assays, we're currently working on the prediction models to assess potency so that you can determine if it's a weak, moderate, or strong sensitizer and can formulate accordingly. Another challenge is the availability of methods. Having labs that will adopt these methods and that you can use them routinely. And of course, cost. We certainly don't want to have these assays become so cost prohibitive that individuals are not able to use them. Lastly, I would just like to once again acknowledge all the organizers for the meeting. Thank you so much for inviting me, and all of my collaborators on this project thus far. We've had many individuals that have been very critical in IIBS's work with skin sensitization. Okay, so that wraps it for me, and I'd be happy to take any questions.
Algunas de la participación como ponente en el segundo encuentro internacional de Querétaro. Ahora